Get ready to enter a brave new world with your host, Vasant Dhar.
Brave New World is sponsored by the Fubon Center for Technology, Business, and Innovation at the NYU Stern School of Business. For more color on the podcast and additional commentary, please subscribe to my newsletter at vasantdhar.substack.com. Hello, and welcome to Brave New World. My guest today is Luca Turin, who I consider quote-unquote Mr. Smell. I became familiar with Luca several years ago when I recorded an episode with Alex Wiltsko, CEO of Osmo.ai. Alex mentioned that Luca's book on perfumes spurred his curiosity about smell and sent him into his journey in olfaction. Luca is a biophysicist, author, fragrance collector, and critic. He refers to himself as an interdisciplinary trespasser and battered survivor of 50 years in academia. Luca, welcome to Brave New World. Delighted to have you on the show.
Well, same here. Thank you for having me. This is absolutely great.
So, I've been wanting to talk to you for some time because you're a one-of-a-kind guy and you've got a unique theory about smell. But let's start with just about yourself. Tell us something about yourself, beginning with your name, which sounds Italian.
It is.
And here we are in London, and you don't sound Italian. Yeah. I'm trying to place your accent. But tell us about yourself and what got you into smell.
So first of all, I am Italian. I actually have an Italian passport. And the reason I don't sound Italian, and I'm glad you asked, is because when I first came to this country in the late 60s, I was faced with the choice of accent. Okay, what accent? You come to Britain as a foreigner, if you're good at it, I'm pretty good at accents. So if you're good at it, what accent are you going to pick, right? Mm-hmm. Because in England, it's X, Y, and Z, right? It's geographic and also historical, diachronic, and then class,
right?
Caste,
right.
So to speak.
Yeah.
You don't want to play that game. I mean, if you could win, it might be worth playing, but you can't.
Yeah.
Okay.
Yeah.
And so I decided to speak American, so I would be basically outside the reckoning of it, and it's sort of stuck with me. So that's why I sound.
I was curious about your accent.
Vaguely mid-Atlantic. I'm shooting for a sort of 1940s movie star. But failing anyway. But I am Italian. I was brought up in France, went to French school, and came here. Came to London to study at UCL. My father was a professor here at UCL, which meant that we. I think for 3 years of study, I paid £10, which my father reckoned to be reasonable value for money. And I came here and I stayed for my undergraduate, graduate, and one postdoc. And then I moved to France for a decade and then came back.
And what did you do in your undergraduate and graduate studies?
I was trained as an electrophysiologist. So meaning the sorts of people who put electrodes in brains or in cells to measure electrical activity. And I did my PhD on junctions on a specialized structure between cells that allows communication between the inside of one cell and the inside of another. And at that point, what happened was there was a revolution in electrophysiology, which basically a technique called patch clamp was invented. And patch clamp is a very high magnification technique. You see single proteins doing their job. And patch clamp has hugely advanced the field, but it wasn't my cup of tea. And so I decided to try other things. And so I did some other work, also electrophysiology, but rather unconventional stuff. And then I got interested in smell.
Is that when you were in France?
Yes. So the smell thing, I've always been vaguely interested in smell, but then gradually, so two things happened. During the 80s, I started collecting fragrance because I had a friend who was an antique dealer in Belgium, and she used to come down to France to where I worked in southern France on holidays. And she would always bring me a ton of perfumes that she bought on the, in those days, they were cheap. Mm-hmm. You could buy amazing vintage stuff for like $5. So she brought me dozens of these, and I started sort of collecting them. And I discovered at that point that I had some sort of brain deformity that made it easy for me to relate to a fragrance. The way I describe it is, a fragrance is not a smell. It's a chemical poem that's been written by somebody, the perfumer, translated into this stuff that's in the bottle, and then you decode it. So I always say it's a message in a bottle. And I found it absolutely spontaneous to read the message in words. I'm not saying it's the only way to read it, because of course even poems can be read in a million different ways. But at any rate, I had this sort of inborn ability to spontaneously say, "Oh, this is what this perfume says to me."
And how did you describe that when you say it says to me? What sort of language do you use?
Because of this, I wrote several perfume guides. So the language is that a perfume can play on a million different levels, but it can suggest a space, an image, a sort of an intent, a literary intent, a story, a mood, a tune, an orchestration. Often my analogies go towards music, because I'm, interested in classical music. And so there's an awful lot of music references in what I write. But it's basically, it's like you're sort of like a wind chime, the perfumed air goes by and you hear a sort of a tinkling, and that's when you describe it. So that's what I did. So what happened was I had this collection of perfumes, and I went to the US, and in the US I could buy a lot of very cheap, older fragrances. As it happened, nobody cottoned on to this. This was the early 90s. I used to bore my friends with my descriptions of perfume until a friend of mine said, "Look, dude, just write a book, okay, and get it out of your system." And so I wrote in French a short perfume guide with only a couple of hundred perfumes in it, which was picked up by arguably the worst publisher in all of France, who was only interested in publishing because they were a bookbinder. They wanted to be present at the book fairs. And so they had some pretext books. Anyway, they published the book. And what happened with that book was, only perfumers really read it. Only people in the industry read it. And they were convinced that I was one of them under a pseudonym. Okay.
And why is that?
Because no one else gave a damn about perfume in those days, except the people actually making them. So it was kind of odd to them that I was passing judgment on their work, and they thought, it must be one of us.
Yeah.
And when they found out it wasn't one of them, I made friends with these people, and I had access to the labs and to all the materials, because in those days it was really difficult to get hold of pure.
And now, when you say those days, you're talking about the
early 90s.
Early 90s. Yeah. Okay. Mm-hmm.
In those days, you couldn't get hold of small quantities of aroma chemicals. They were, they were sold by the big companies. There was nothing much at the retail level. And so I had a chance to go into the companies and smell the stuff. And what happened was, in the meantime, I had been reading up about how smell works, and it didn't really make much sense. What I was reading didn't make a lot of sense. But there was one aspect of it that really bothered me, and which I think is crucial, and that most people don't really think about very much. And that is the fact that chemists have a particular view of smell. Not perfumers, not chemists, not psychologists, chemists. And the way they describe smells is really interesting, because a chemist will tell you something is ethereal, nitrilic, sulfuraceous, etc. What they're talking about is functional groups. Okay, so a functional group is a small collection of atoms, typically 2 or sometimes 3, which is the most reactive part of a molecule generally. Mm-hmm. And you can attach that hook, so to speak, to any sort of molecule. And what's very striking and chemists, of course, know perfectly well, is that no matter what the rest of the molecule is, you'll smell the functional group in it. So any molecule that has a C triple bond N or an SH will smell respectively nitrilic and sulfuration.
So what does nitrilic smell like? Just
it's very easy.
How would you describe it?
So nitrilic is a metallic smell. So, for example, when you put a CN group on the cucumber aldehyde instead of the aldehyde, you get a metallic cucumber. You can get metallic lemon. So for the people who are old enough to remember this, lemon-scented stove cleaners used to be a lemon nitrile. So they were lemony, but metallic lemon. So chemists are fully aware that you can smell functional groups in any context. And when you think about this fact in the context of molecular recognition, how are you going to detect that? It's really hard to understand because a functional group makes, let's say, one bond hydrogen, but it's a very small thing. You can only make one bond with whatever receptor. How does the receptor know that that bond is made to a nitrile rather than a sulfide or whatever? And this was going around in my head, and I was aware of the existence of the vibrational theories of smell, which had been proposed in the late 20s and early 30s, and then proposed again in the 60s. And they made sense, to some extent, except for the fact that there was no mechanism. None. So you can say, well, the nose detects functional groups because of their weird vibrations, because the fact is each functional group has a very specific vibrational signature. In other words, because it's a small bit of the molecule, it vibrates at a specific single frequency typically, and those frequencies are in a range where they're not overlapping with anything else. And so you could, in principle, and that's how chemists used to do it, you'd put an unknown molecule in a spectrometer, and you'd look in that region and you'd say, aha, this dude has a nitrile, a thiol, or whatever. So it made sense from an empirical standpoint, but it made no sense at all from a mechanistic standpoint, because, if you look up someone's nose, there's no spectrometer up there, right? Spectrometers of optics, prisms, detectors, whatever. So it made no sense. And people had given this theory, I think, a remarkable amount of attention, considering there was no mechanism. Because in those days, remember, the olfactory receptors were identified in 91. Okay, so before 1991, it was a free-for-all as to what the mechanism might be.
You're talking about like Axel and Buck?
That's right, Axel and Buck. That's 91.
Yeah.
So until 91, it could be anything. And so every biophysicist worth a damn had some crazy idea about olfaction. And then after 91, it had to go through the receptors. And there was no obvious way that receptors might be dragooned into measuring vibrations. Okay, there was no obvious way to do that with a protein. And then one day I came across this paper in the Review of Scientific Instruments that explained that, in fact, it had nothing to do with olfaction. It was a purely technological paper that described a type of spectroscope, which is essentially a nanotechnology thing, a device involving electron transfer across a short distance.
So say more here, because I suspect we'll lose some listeners here because there's a lot of jargon going around here.
Right.
So a spectroscope actually just measures the frequencies, the distribution of frequencies in something.
Right. So the idea is this. You can engineer a device which consists of a small gap between two metal electrodes. And you put in there a molecule that you're interested in. And then you vary the voltage across the gap by changing the voltage on one side, and you measure how much current flows. And the current can flow in two ways.
And this is with or without a molecule?
With or without a molecule. That's right. So without a molecule, you get an absolutely linear current flow.
Right.
There's no deviations from linearity.
Right.
With a molecule, you get little bumps on the linear curve. And those bumps happen when you essentially, instead of having just one way to get across the gap. You have two ways. You have one straight across and one second way that involves making the molecule vibrate at a particular energy and then going across. So these bumps on the curve indicate you can process the data in such a way that you get what actually the vibrations of the molecule.
Okay. Now, does this have anything to do with resonance?
Yes, absolutely. It's
Is it the same concept?
That's right, resonant tunneling.
Okay.
So when I read that paper, what really struck me was this. This was intrinsically a nanoscale thing. You could make this phenomenon of electrons jumping across the gap only works when the gap is 1 nanometer across.
Yeah, really
Maximum,
tiny.
Yeah. So this is protein scale, okay? Number one. Number two, electrons are flowing through biological systems all the time. As you and I sit here, we have something like 70 amps of electrons flowing in our mitochondria site to oxygen. Oxygen is being turned into water by an electrochemical reaction. So it's not as if electrons are scarce in biology. They just haven't been used conceptually for anything other than reducing oxygen. So I thought, look, we have the elements. We have a nanoscale device. We have electrons flowing. Imagine you have a protein which has a pocket in it in which you can put a smelly molecule and then make electrons flow across the molecule in the protein. You could build a spectroscope in parts. So it would be like color vision. One type of receptor takes care of one particular part of the vibrational spectrum, and the other, so it'd be like red, green, blue. Okay. And the striking thing about the red, green, blue thing is that we've got these three poorly tuned, really, receptors. I mean,
you mean in vision?
In vision,
yeah.
In the RGB, they have broad resonances, and yet we can tell apart, I don't know how many, 15, 20,000 colors.
Yeah.
So this had it was attractive because it was plausible on the physics side, plausible in terms of discrimination, and also it had that wonderful thing, the unity of the senses. You know, if you put your German philosopher hat on, for a minute, you think, okay, we've got three spectral senses: hearing, color vision, smell. And they measure, respectively, compressional waves in air, photons, electromagnetic waves, and vibrational waves. There's something wonderfully symmetric about it. That doesn't mean it's true.
Yeah.
Many mistakes in science have been made.
No, I mean, it's a theory.
Yeah, but it's a nice idea. Anyway, so I started gathering evidence that would support this. But the first thing I did...
Now, when was this? Was it 90s?
92.
Okay.
92, 93.
Okay.
I was at UCL. Because I was into smell and I was talking to all these people, 93, 94, I was talking to all these people in the perfume world, and I really started to focus on how pure molecules actually smell. What was interesting to me was that there was one smell character, which was really unique to a particular vibration. Okay? And this was the sulfur, the rotten egg, as they call it. I don't know how many people these days have smelled a rotten egg because they get...
You don't see them, come across them often enough.
No, they're very rare enough. But the rotten egg character was very special because if you look in terms of vibrations, it's a very unique vibration. And
now, when you say a unique vibration,
the frequency
of what? Of which?
Of the SH bond.
Okay.
So sulfur, hydrogen, a stretch vibration, just a very simple.
So hydrogen sulfide.
Hydrogen sulfide.
Typical, yeah. Mm-hmm.
And I decided to try and see if there was something out there that had the same vibration, because the prediction would be absolutely that it should smell of sulfur, of SH. And eventually, after much messing around, I mean, today I could probably do it in 5 minutes, but in those days it took me like months. I found a class of compounds called boranes, it's not SH, it's BH. And it turns out...
That's what, boron and hydrogen?
Boron, hydrogen. So boron hydrides, boron hydrogen compound. And I found that they had a reputation of smelling awful, which was already kind of interesting because people hate the SH smell. It's the smell of decay.
Yeah.
And I spent a few weeks looking to buy a small quantity of a borane to smell, because they should all have that smell. And eventually I found one, amazingly, that I could order, and it would come the next day, 1 gram of this stuff. Most boranes are explosive and spontaneously combust and all manner of, and they're toxic.
Yeah.
So this dude was okay, stable, and reasonably non-toxic and so on. And I ordered it, and it smelled of sulfur. It smelled of boiled onion, which is a sulfur molecule. And that was it. As far as I was concerned at that point, there was simply no way this was going to be a coincidence. That the only thing in the world that actually smelled like sulfur was completely unrelated chemically and only had a vibration in common.
Now, when you say it had a vibrational equivalent,
the stretch frequency of BH is the same as SH, and nothing else in the entire planet or universe for that matter.
And that's known?
Yeah.
That the frequency is the same?
You measure. And at that point, I thought, okay, now how do we make this work? And I wrote a paper in 96 explaining this idea. And 30 years later, we're still not exactly there, but there's still, the debate is still going on.
In fact, I want to read a line. I read your 96 paper, right? So I want to read something from your abstract that you wrote, and then I want to build on that to discuss some of the alternative theories in smell, right? So here's what you say. You say the mechanism by which receptors detect odorants and thus the molecular basis of odor remain unclear. Structure odor theories tend to reflect one or the other line of evidence. Quote-unquote, functional group theories emphasize the analytical view but do not provide a mechanism by which individual groups can be detected regardless of molecular shape. Theories based exclusively on shape enjoy some success within specific odorant groups, but do not account for the striking chemical group regularities, nor do they account for the fact that very different structures give similar odors. End your abstract by saying the evidence presented here suggests instead that olfaction, like color vision and hearing, is a spectral sense. So let's dig into this because there's a lot in this paragraph. What are the major competing theories of olfaction? Or you still haven't completely laid it out. You've sort of provided a basis for it, which is that it's based on vibrations. But I don't think you would rule out structure entirely.
No, of course not.
Right? So that's one view. What are the other major theories of smell, including this sort of more recent sort of principal odor map?
Right.
So why don't you describe them, and then we'll talk about why we should believe one or the other.
Right. So first of all, let's put smell in context. It is a branch of pharmacology, so to speak, because if you have smell receptors, and we do, and they are G protein coupled receptors, which is a particular subclass of receptors. So, and G protein coupled receptors are everywhere in the brain, and are the target for many, many drugs of great medical importance. So it's an area in which there's a tremendous amount of research completely outside the fragrance industry.
And now just for listeners, the G protein receptors are the ones that are in the nose, in the
nose, in the olfactory epithelium.
Yeah. Okay. Right. Yeah.
So what's different about smell is that unlike, say, the serotonin receptors, where you have, I don't know, 6 or 10 types, and the dopamine receptors, in which you have maybe 5 or 6 or whatever. In smell, you have 400 or 500 different receptors. And we'll come back to that in a second. But,
compared to 3 in vision. Keep that in mind.
Yeah, that's right. So where the shape comes in is that since Ehrlich in the early 20th century, Paul Ehrlich, the idea of the magic bullet, which turned into what's called the lock and key theory, is the dominant theory of molecular recognition in general, be it antibodies, be it drugs, it doesn't matter. No matter what the size of the thing is, the general idea is it needs to bind specifically to a receptor, and once in the receptor, either turn it on or turn it off, because there are some drugs that turn on receptors, agonists, and some that turn off, antagonists.
Right.
And there are a variety of things in between. So this is the absolutely dominant view. But there's an interesting angle to that, which is that as any pharmacologist or pharmaceutical chemist knows, you can have a drug which binds very tightly or not so tightly, let's say, to a receptor. And that's what's called affinity. The affinity of a drug for a receptor is determined, I would say 100%, by its shape. And by shape, one means the ability that the drug has to make bonds, weak bonds, not covalent bonds, not permanent ones, but weak bonds to the receptor. So you have a geometric arrangement of groups on the molecule that basically fit correctly in the receptor pocket and so on. And that ensures that when you give the drug, it's going to go to one receptor. So when you take a serotonin antagonist, like I suppose LSD or something, it does that. And there's another related molecule that might cause uterine contractions.
Yeah.
But those receptors are different in structure, not hugely different, but different enough that only one molecule will bind to them. Okay. Now that works really well for the binding, for the affinity. But then there's another step to any receptor action, which is what's called efficacy. Meaning, okay, the molecule's bound. What does it do next? Does it turn the receptor on or off? And that's a much more mysterious thing. Because to this day, particularly with G protein coupled receptors, there is no general theory of efficacy. So you can tell by molecular modeling and with all manner of chemical techniques and computational ones that a molecule will or should or does bind very tightly to a receptor because it makes this without interactions. And now we have the structures of the receptor, so you can actually fit them. But you don't know. You simply just do not know what the molecule will do. And when people try and simulate this with molecular dynamics, you have a complete model of a protein where with each atom and the interactions between the atoms are modeled by very simplified force fields and so on and so forth. And you put the drug in, let's say, in a computer experiment, and you run the thing, and you know that drug turns on the receptor in practice. Well, does it turn on the computer model of the receptor? The answer is no. And there could be two reasons for that. One is your simulation doesn't last long enough for that event to happen, and that's commonly what is believed. In other words, when you do a molecular dynamics simulation of a protein, the simulation time never reaches a millisecond. You're stuck in the, I think, whatever, hundreds of nanoseconds, maybe 100 nanoseconds.
And why is that?
Because it's just a lot of, your increment, your time step would be, let's say, 1/10 of a femtosecond. And so to get to a millisecond would take too much computer time. Right now, limitations. So no one has managed, that I know, if anybody who's listening has a counterexample, I'd love to hear it, but no one seems to have managed to simulate adequately on a G protein couple of receptors an agonist or an antagonist and get one drug to turn the receptor on and the other to turn it off. Okay? So, bottom line is, what happens after binding is still a mystery. And I believe that's the crucial mystery. You say over the years, in my own mind and due to debate and actually controversy with colleagues, I've come to the conclusion that there's a central problem, which is not just for olfaction, which is a central problem of G protein coupled receptors, which is there's something missing in the mechanism by which a drug or an odorant, for that matter, turns them on. Okay? Now, I want to mention something which is very specific to odorants. There's an additional problem with smelling molecules, which is that the reason that you smell them is because they evaporate from whatever.
They're volatile.
They're volatile. And the reason they're volatile is they don't make very strong bonds to each other or to the solution or to the substrate, to your skin, to a piece of paper or anything. They're actually very slippery molecules. They typically don't have the kinds of groups on them that enable a drug to make bonds to a receptor. In other words, they're lipid soluble. They're soluble in oil, which is why perfumes are often oily.
Mm-hmm.
And they're not very water soluble. And if you look at it, most drugs, not all, but most drugs are fairly water soluble because the pockets of the receptors are, after all, facing the water thing. So smelly molecules do not have the groups on them that would enable a very specific interaction with a receptor. They will dissolve, and this is being shown, in almost any oily pocket of any protein, provided it has the right size. So actually, there's an additional problem, if you're thinking in terms of receptors, with odorants, which is that they don't have what it takes to be very specific. And furthermore, their energy of interaction with the molecule is weak, is low, because they don't make these strong, medium strong hydrogen bonds with things that normally you'd expect. The general reckoning is that.
So you're saying that this is a property of volatile.
Yeah, that's right. So you have, it's compounded. So the problem of what makes a molecule efficacious in turning on a receptor or turning it off is compounded when you deal with odorants by the fact that they're slippery guys, very slippery. So, of course, if you're measuring their vibrations, doesn't matter. I mean, the thing is there. You measure the vibrations. The mechanism by which you probe the molecule and turn on the receptor, one size fits all in this particular instance. Anyway, so this is where we are. So, to sum up where we are now, okay, as far as my theory is concerned, I think that the strongest objection to the vibrational theory, and one which I very much take to heart and which was put forward by a colleague of mine, is that why would evolution have made olfactory receptors so different from all the other G-protein coupled receptors? Why would evolution have engineered an electronic mechanism for that class of receptors alone? It does seem kind of odd, frankly. And my answer to that was, for the longest time, was, I don't know. And now I believe it's not a unique mechanism. I think all G-protein coupled receptors work by an electronic mechanism, and it's probably much easier to test in the normal G-protein coupled receptors like serotonin or whatever. And we're actually working on some experiments in that direction. So this is where we are. The biggest problem was that every time I spoke to a G-protein coupled receptor person, specialist in GPCRs, they agreed with me.
About what?
That there's a problem with the transduction, what's called transduction, which is how do you turn a binding event into an activation. Okay. The people who are slightly further away from the coal face don't realize that that's a major and largely unspoken problem in GPCR pharmacology. So I think things are coming together now, but I don't think that I'm going to prove that olfaction works that way. I think, if anything, there's a decent chance that one might prove that some other GPCR works with an electronic mechanism, not necessarily to do with vibrations, just a straight up transfer of electrons across the drug.
So let's back up a little bit here, because you said a lot. And I just want to make sure that I understand the competing theories. So the lock and key theory is purely structure-based, right? And I think you point out somewhere, and we'll introduce a little chemistry here. I mean, I had to go back and refresh my memory about isotopes and isomers and isotopomers, which is the basis for your theory, right? Yeah. Because what you point out, I'm going to try and express this simply and tell me if I'm right, is that if it were purely structure-based, then two isotopomers, okay, so isotopes are where you substitute an atom with something else, or let's say you add a neutron, right? But it doesn't change the protons, the electrons, it just changes the mass, right? And what you claim is that they smell different because one's heavier and it has a different sort of vibrational frequency, right?
Yes.
And so you're saying that a purely structure-based theory fails on that, so it can't account for that, right? And so you propose this sort of vibration mechanism in addition to structure, right? So you're saying that structure still matters, and in fact, when I sort of dug into this, the structure of these receptors is that they tend to be chiral.
Yes, of course.
Which means that the orientation matters.
So first of all, in the intervening years, what happened was I tested with my colleagues in Greece in the lab where I was working at the Fleming Institute in Athens. My colleague Makis Skoulakis. We did two types of experiments. First of all, we trained fruit flies to distinguish isotopes, to tell isotopes apart. And they are very good at it.
And they can.
Oh, yeah. Much better than we are.
Yeah.
And so there were various types of experiments where basically they can tell isotopes apart. Yeah. And that's fine. And then you can fool them by when you do, for example, so typically the experiment is you have a molecule, as you said, which has, let's say, 10 carbons and 20 something hydrogens.
Yeah.
You replace all the hydrogens, or most of them, with deuterium, which is heavy hydrogen. Yeah. From a structural standpoint, they're essentially identical. Yeah. From a vibrational standpoint, they're completely different because every mass has changed. Yeah. Can the flies tell them apart? Yes. Can you fool the flies? Because the vibration frequency of the deuterium looks a little bit like the vibrational frequency of the nitrile that we talked about earlier, the C triple bond N. Mm-hmm. So can you fool them into thinking there's deuterium by putting a bit of nitrile in there? And the answer is yes. Okay, that's another nice experiment. And then we decided to go whole hog. Go ahead.
And I was going to say, when you say, can you fool them, you mean you still want them to distinguish between...
Well, you do an experiment where you train them to avoid a deuterium molecule.
I see. Okay.
And then you instead of the deuterium, you put a nitrile.
And they're still
avoided. They think, ooh, this thing doesn't smell right to me.
Right.
Okay.
Right.
But then we decided to take the next step and to do with humans, because the early experiments with deuterated molecules, heavy versions of odorants, were really inconclusive, because the deuterated molecules that you have access to, that you can buy, are typically solvents for nuclear magnetic resonance. So they're a very small subset. Yeah, you can buy them, but they're small solvents. And when you test them on humans, you find that the differences are small. Okay, the difference between, and it's not just small, elusive. It's there for a minute and then you don't smell it anymore, that kind of stuff. So I had this, well, based on my perfumery experience at that point, I thought, here's what we can do. We can take a musk, which is at the high end of the odorant range. The musks have typically 18 carbons. And
so quite heavy.
Big, heavy things. I mean, they're still small compared to the receptor. Yeah. But they're at the biggest, roughly speaking, nothing with more than 18 carbons has a smell. Mm-hmm. Right.
It's not volatile enough.
No, it doesn't fit in the receptor. It's plenty volatile enough.
I see.
It's plenty. I mean, it's going to be less volatile than 19 carbons. Yeah. If you have 20 carbons less, but still, you should be able to smell it. You don't. Mm-hmm. So we took an 18 carbon musk and took advantage of a newly developed, reasonably mild, low temperature technique to replace every hydrogen with a deuterium. So you take you buy from a fragrance company a pure 18 carbon musk. It smells fantastic, by the way. And you treat it at lowish temperature with a rhodium catalyst, and you replace every hydrogen with deuterium. And it stays pure. There are no side. I mean, you can purify it afterwards, and we did. But basically, you've got yourself an essentially 1-for-1 replacement of hydrogen for deuterium. with deuterium. And the answer is, does it smell different? And the question is, does it smell different? And the answer is yes, hugely different. As in, not a pleasant musk smell, but a burnt candle sort of nasty smell, okay? Which is very, very interesting. And not only that, but my friend Marcus, who's completely anosmic, he cannot smell the normal one, the hydrogen smell, musk. He smells the deuterium musk intensely. And we published that. And okay, so what happened was not as much of a shift. I mean, the people who believed that this might be true were comforted, and the people who thought it was safe tried to ignore it. Really, yeah. But then something very interesting happened, which was that some colleagues at the Rockefeller University published a big study in PNAS, I think it was.
Who was this?
Leslie Vosshall. And found that in tissue cultured cells in which the receptors had been added by engineering, there was not much of a difference between isotopes and that kind of thing. So
you mean humans couldn't tell?
No, the cells themselves, if you tested their response to odorants, so this was in vitro. It was objective measurements. They found nothing much. Okay. So they said, well, this is the state of the art technique to measure activity of receptors, and it shows nothing. Okay. And then a big editorial in PNAS.
But hang on, let's back up here. How do you do this in vitro?
So you take a cell line, a standard established tissue culture cell line, meaning they're immortal. They grow easily, whatever. It could be an insect line or a particular kidney line. There are many that are commonly used. And you insert into the machinery of the cell, the gene that makes a particular smell receptor. And then it's there because when you put an odorant on, the cell responds. You can measure the response. If whatever happens inside the cell, you can tell it's responding. And then you can test different odorants and see what happens. And they found nothing much really with isotopes. And they concluded from this that given that this was the state of the art, method for measuring receptor activity, and given that they found nothing, the vibrational theory must therefore be wrong. And okay, it's a data point on the negative side. This was accompanied by an extraordinary think piece, editorial written by Leslie Vosshall herself, saying basically, let's put this theory permanently and forever to rest, which I think is slightly over-egged because no single experiment of that particular type could ever achieve that. I mean, it's everything in science is a little bit more complicated than that, especially fusing tissue culture cells and so forth. Anyway, a few years later, a group in New York, in fact, showed that if you used actually olfactory neurons, primary culture, in other words, you take the neurons from a rat olfactory epithelium and you dissociate them into tissue culture, and then you test the odorants, you find that they do respond differentially to isotopes. And it was kind of interesting because not all of them did, but a smallish fraction of the neurons responded completely differently to octanal, I think it was, and deuterated octanal. And to me, it's like, the parallel that I make is that you say pigs don't fly, okay? And then you find one pig streaking through outside your window, okay? So pigs fly. There's a qualitative element to such an experiment, which is, I don't care how few of the receptors do it.
There is one.
It's like pigs fly, just rarely. But there's a tendency among biologists to say, on average, pigs don't fly. Which is completely absurd, right? Big difference between some pigs fly and no pigs fly. I got the paper to referee. It was sent to me. And I looked at the discussion and it said, well, this could be due to a hypersensitivity of receptors to isotope composition. And my reply as a referee was, that's not an explanation. You're just restating the result in some
as hypersensitivity.
Yeah, it's like that's what your doctor would tell you. That's not a scientist speaking.
Right.
And my comments, and I signed them, I wasn't going to pretend to be anonymous. I was like, dude, you have evidence directly in support of a vibrational mechanism, and you're telling me it's a hypersensitivity without...
But hang on, does that support the vibrational mechanism?
Yes.
Or could there be other reasons for it?
Not at that level. You see, there could be, if you find a 1% difference in receptor responses, you can always argue that the diffusion constant or the binding is slightly different.
I guess the reason I'm asking this is because the main sort of criticism of the vibration theory is that there's no direct evidence.
That's nonsense.
Is there?
No. Look, how do you explain the fly results on isotopes, the musk smell difference?
I don't know, but clearly we're not like observing things vibrating.
No, I agree with you.
Yeah.
And that's really the missing link.
Yeah.
But I think there's a very basic difference between a small effect and a big effect. A small effect, many, many small effects, isotope differences, could be explained by small differences in diffusion constant, binding constants, hydrogen bond, whatever. You plenty.
Yeah.
But when you have a primary olfactory neuron dissociated in a fresh culture that does not respond at all to normal octanol and responds strongly to deuterated octanol, no amount of messing around with second order effects will explain that. It has to be vibrations. There's no other explanation. And if you show this to any physical chemist, they'll say that discussion doesn't make any sense. You haven't explained it.
But why do you say it has to be vibrations? Why couldn't it be something else? I don't know. I'm just saying.
No, I'm totally open. If someone comes to me and says that actually the neurons are doing nuclear magnetic resonance, and they can easily tell whether the nucleus of hydrogen is deuterated or not, then I'm a happy guy. I'm wrong, but I'm happy. Okay? But the fact is there's no mechanism to do that. There's no mechanism, no known mechanism. If somebody figures one out tomorrow, terrific. But as we stand, you cannot explain it any other way. A large effect. Okay. So that's where we are in a way.
Okay. So, what about, the first time I heard of you was from Alex Wilsko, who's the CEO of Osmo.ai, smell company. And he's got this theory of a principal odor map. Like, how does that fit into shape, vibration? Is it completely orthogonal or is it related? So my understanding of it is that he essentially maps molecules and maybe some auxiliary information into a vector. Right? And the idea is that these vectors are learned, and vectors that are close to each other correspond to things that smell similar. And if they're far apart, then they smell very different. It's interesting theory. Like, where does that stand, relative to shape versus vibration?
Well,
is it different? Is it unifying?
So, first of all, let me say, an odor map is what we're all looking for. Okay. The thing is, what are the axes? Because it's really a three-dimensional or n-dimensional map. And the question is, what are the coordinates?
Yeah.
So the way I see it is that Alex Wilsko's work is an extension of what used to be called quantitative structure-activity relations. So the people have been doing this in the drugs world for a long time. And the way that they did it was you have, let's say, you have 100 drugs and you know how powerful they are, what they do, and then you statistically analyze the relationship between their potency and any number of variables. You can put in anything. And in fact, they really did put in anything. Like they put in things like topological indices of the molecule. In other words, the branching, graph theory stuff, whatever, which there's no chance in hell that a receptor would actually be a graph theorist, but never mind. And they got these clouds and they looked at principal components and that kind of stuff to try and figure out which properties correlated best, which of the long list of things that you put in correlated best with whatever they were interested in. And this has been going on for years. I've never fully understood how much benefit was gained by drugs companies from this. As an academic exercise, it always struck me as highly suspect because, first of all, the criteria that were used were so unbiological, unphysical. There's, if you say this molecule has a branching number or a graph theory number of 7 or something, it's like, okay, how is that going to be detected by a biological system? Come on, But secondly, the worst thing, the most dangerous thing was that they tend to ignore outliers. Because it's a statistical technique that's focused on the shape of the cloud and the principal components, the thinking there, when you're dealing with principal component analysis, is that you tend to say, okay, well, those two guys that are over here and over there that don't fit the cloud are somehow less important. Okay? And I've always taken the very old-fashioned view that outliers is where the information is. That they tell you more than or at least as much as the cloud. So to come to the odor map, Wiltshire, if you look at that paper, it's the logical evolution of quantitative structure-activity relations via Hoehn maps, the unfolding of relationship to AI. And in QSAR, in quantitative structure-activity relations, you had the principal components, which are often difficult to interpret. It's a sort of 0.2 of this plus 0.3 of that. What the hell does that mean? Okay. In the Hoehn maps, you have relationships that you unfold them, but it's sometimes quite hard to figure out what the map is mapping. In machine learning, you really don't have much information as to what's going on inside the beast. So that's my criticism of this sort of approach in general, is that ultimately with a machine learning thing, you don't know really what the network has learned. And it could be much more vibration or less vibration than you suspect, because it's not going to tell you how it arrived at those weights. Now, specifically on the science paper on the principal odor map from 3 years ago, there are a couple of things. It's really not my job to criticize colleagues who are doing what I think is super interesting.
No, we're just sort of comparing theories.
So the way I see it is this. Number one, they failed completely on musks. And musks really are what I was talking about earlier, the outlier.
When you say they failed completely on musks, meaning what?
Because there are no structure-activity relations for musks. Musks, historically, from the 1880s, when the first synthetic musks were discovered, there's been a succession of molecular...
So do you mean that you give it a musk molecule and it predicts that it smells like sulfur?
It has no idea. No, sulfur, no.
But what does it predict?
I don't know, because I haven't looked at the detail of their data, but they say that...
It gets it wrong.
Yeah, it gets it wrong. Okay. And that's because musks are extraordinarily diverse structurally and have very, very similar smell. Okay. Fine. Number two, they throw away fully 20% of their odorants they eliminate them from the assay because of various considerations that I don't fully understand, but it makes me slightly uneasy, because if you throw away 20% of your database at some point, what are you throwing away? Okay. And thirdly, the perennial problem with all neural networks that you don't know what the weights are doing. It's a black box.
Right.
Okay. This said, what they've got is impressive. I just don't know how to interpret it in terms of a mechanism.
But it seems like an interesting sort of first order approximation,
absolutely.
Right? Going from molecules to smell.
No, absolutely. Look, the intent is absolutely logical and straight up, and I'm very excited that they're doing this. I will say this recently. So they're doing this on mouse in the piriform cortex, right? Mm-hmm. Recently, the most extraordinary thing happened, which is that a bunch of kids, and I mean kids, I'm old enough to call anybody under 25 a kid. These kids in California pointed an ultrasound beam, if you attach an ultrasound transducer to the head, you can stimulate neurons that are exposed to the ultrasound. And if your ultrasound beam focuses somewhere inside the head, not at the surface, you can stimulate deeper structures. And stimulate is, by the way, I'm saying stimulate as a general term. Yeah. Actually, it's not clear what the hell's going on. And I personally have always felt very nervous about this, using ultrasound. But anyway, they did it. And what they did, I think largely because they had an ultrasound stimulator and time on their hands, they stimulated the olfactory bulb. The olfactory bulb is a very weird thing that sticks out horizontally from the base of the brain into the nose, and is about 12 millimeters long and 5 millimeters wide. There's two of them, right? There's one on each side.
Yeah.
And they're shining ultrasound on the bulb, and they get a smell sensation. Okay? This was last December. And not only that, but when you change the exact position of the ultrasound beam, the sensations change in a reproducible way between people. Okay, so there's clearly an odor map in the bulb, which needs exploring. And I don't know whether as soon as I have some cash, I'm going to get one of these ultrasound machines and start fooling around.
On yourself?
Yeah. I wouldn't probably get approval to fool on anyone else.
Yeah.
So, but let's think about this for a second. If you have an odor map in the olfactory bulb, it's going to be fairly straightforward to figure out whether it's vibrational or not. Okay, because you could predict that in some part you'll get a garbage smell, the SH. Okay, you'll get a metallic smell, you'll get an ether, you'll get the functional groups. Basically,
but you'd have to know which part is being stimulated.
Yes,
right,
in fact, the beam, if you do it right, and it's fairly low power, if you do it right, it seems like they can stimulate, let's say, 1 millimeter or 2 millimeters of the total thing.
Yeah, but you can't see which it is.
No, you don't know.
Yeah,
you could if you combined it with MRI, but that gets complicated.
Yeah.
Okay, but anyway, these kids have hit upon a truly amazing thing, because this is something that people don't realize, that it's never been possible to stimulate olfaction by anything other than molecules. Mm-hmm. So, for example, if I get a bang on the eye, I get a flash of light because the neurons in the retina are going, yow! Okay?
Yeah.
If I put magnetic stimulation on the back of the head, I see lights in the visual field. Mm-hmm. Okay? So, you can get sensation at both ends. If I put a cochlear implant, I hear. If I stimulate the auditory cortex, I get clicks or whatever. Nothing has ever worked. I've actually spoken to people who spent years putting electrodes up people's noses and trying to stimulate the olfactory neurons. And goes precisely nothing.
Yeah.
Now these kids, and when I spoke to Lev Chizov, he's a young guy who's into ultrasound stuff. First of all, he looked about 17. He's 23. He's a classic Russian genius, mathematical Olympiad, École Polytechnique in France, moves to Stanford or whatever, and he's now, doing amazing things. He explained to me that they'd done it on the ear, on the auditory cortex. And then they said, what else can we do? And they did it on the bulb. And they just chanced upon this astonishing.
Have they published the result?
No, it's on their blog, so far as I know so far. I mean, it hasn't been published in a science journal. I don't know. I have no idea. I mean, I was in touch with them immediately this came out.
How would you make something like that publishable? How would you do a proper experiment with something like that?
Well, experimental design is really, really...
Would you need lots of people? And
it's hard to say. I mean, you can only tell when the results come in.
Yeah.
But I would say, for example, a basic prediction, in my opinion, would be that it's tonotopic.
It's what?
Tonotopic. So if you look at the other vibrational senses, like in hearing, the cochlea is arranged by frequency.
Uh-huh.
And that map by frequency projects onto the midbrain and onto the cortex. So in other words, if you stimulate adjacent points anywhere along the map, you'll get adjacent tones.
Mm-hmm.
Okay. If olfaction is vibrational, that should apply possibly to the bulbar map. You should, for example, if you find the 2500 and you move one way or the other, one direction or the other, you'll hit the 2100, which is nitrile, which is metallic. So you can make some predictions. And if they come out and you explain to subjects, here's a sulfur molecule, here's a nitrile. When I do this, and you don't, of course, the subject doesn't know what's happening. What do you smell? You could do that. It'd be work, but nothing stops you from doing it. If I may interject at this point, there's an interesting aspect of olfaction, which is that, in my experience, a sizable fraction of people who work on smell do not actually smell things for themselves. The Venn diagram of working on olfaction and having an interest in olfaction as a sense, be it perfume or food or even just smelling aroma chemicals, it's a very small overlap. And this has been a problem because when you talk about vision, you can show, for example, a visual illusion to a lecture hall full of people, as Edwin Land famously did with his amazing color vision stuff. And you've got basically 400 people at the same time seeing roughly the same thing, except those who have a color blindness or something. With smell, if you distribute smelling strips, people get incredibly queasy, they get nervous because they don't trust their sense. They think, many people seem to be of the opinion that whatever they smell is what they smell. Maybe no one else does.
Is that because we don't learn how to smell? Like we learn how to see and hear and read?
Yeah, I don't really know what the origin of that is. Yes, there is definitely a deficiency of empirical.
But we don't even talk about it, right? We don't even talk about smell.
I talk about it all the time. So, anyways, so I think this discovery by Lev Chizhov and his colleagues is really a turning point, and could be a shortcut. Not to the mechanism, by the way. I mean, of course, it doesn't tell you whether receptors detect vibrations by whatever.
Yeah.
But if there was a tonotopic, frequency-related odor map in the olfactory bulb, then I can retire to the Bahamas happily.
So let's segue from theory to application. Like, what do you see as sort of the applications of smell in the next 10 years or so?
Well,
how might it change our lives? And, I'm referring to, the example of Joy Milne, who's hyperosmic and smell Parkinson's with 100% accuracy.
Oh, that kind of stuff,
a decade before it's, detected by conventional methods, right? So clearly we're ignoring some information here that we could be getting about ourselves through olfaction that we're not getting through other modalities.
Right.
Is that something that you're optimistic about?
So let me just tell you a story. I've always been rather skeptical of this stuff, and I absolutely believe that dogs can smell disease.
As can humans.
Yes, and in fact, Chinese medicine trained people in the old days for that. The proper Chinese doctors would smell you.
Yeah.
Now, one thing that really opened my eyes and nose was that some years ago I had muscle spasms to do with some form of sciatica. And I took an anti-spasm medication called tizanidine. And it's an alpha-1 blocker, allegedly only spinal cord, but actually gets into your brain. And after a couple of weeks on the high dose of tizanidine, the spasm went away and I stopped it. And for the next 3 days, I had the most extraordinary experience in my existence, which was hypersensitivity in all senses. Couldn't stand any light, had to shut the shutters and be in darkness in my bedroom. Any sound was incredibly loud, and I could smell things the way I'd never smelt them before. I could smell things like a dog. And at that point, I actually rather felt sorry for dogs because, for example, the towels in the bathroom all smelled rank. The way the badly washed,
right.
They were perfectly well washed, and normally I would never notice.
Yeah.
But I suddenly had for 3 days hyperacusia and hyperosmia, and that really opened my eyes to what we could, in principle, do. I'm not sure how one would induce this, but by God, if you had that, you could probably really get a job as an avalanche dog and demand more money. So I'm completely convinced that any machine, other than a human, who can do that sort of thing, would be hugely useful. I mean, I see in the future that when you do go for bloods, there probably will be a machine, like a very small mass spectrometer of some kind, that will do the same for breath. Or for general odor, and detect metabolism problems and all that. So in terms of diagnostics, yes, and we really should work on that. When it comes to the fragrance and flavor industry, it's a very conservative industry. I mean, the technologies are. Every time people have tried to introduce a very new technology for delivery of odor,
How much innovation is there, by the way, in the fragrance industry? Did you get amazingly new perfumes coming out from time to time?
Yeah, you get amazing molecules. In recent years, the emphasis has been on increasing the power, the strength of odorants, rather than really creating new smells. And the reason you want to increase the strength is because all the environmental concerns are predicated on how many kilos or tons of the stuff you make. So all the allergy concerns have to do with dosage. So making an odorant much stronger means that you put less, and therefore there's going to be fewer allergies, fewer problems, fewer transport costs, fewer everything, fewer environmental costs. So the industry has focused a lot on making more potent odorants, not necessarily with very interesting or novel odor characters. They've also focused rightly on replacing all the odorants that have been sort of banned or restricted because they're allergens with other ones. But in terms of pure novelty, yeah, there's some new stuff, but it's the kind of stuff that would make a perfumer go, wow, not perhaps.
What about in like security, bomb threats or things like that? I mean, at the moment we have dogs or very specialized kinds of sensors. Do you see olfaction going into that area?
Yes, very much. I mean, dogs are, of course, amazing, but they can only be trained to one or two things. So you have cocaine sniffing dogs, explosive sniffing dogs, and they don't do both. and they're high maintenance. You have to have the dog handler and everything. So yes, a machine. I mean, okay. So the machine that detects, the swab thing that they do at security, which detects nitro compounds.
Mm-hmm.
Okay. So a friend of mine, Chris Sheldrake, who's a perfumer, he went to India, and in India, they still sell nitro musks. You can buy nitro musks by the ton in India. They've been essentially phased out in the West. And he was smelling them because he's a fantastic perfumer and he loves the old stuff. And of course, at the airport, because the nitro musks are relatives of TNT that are shitty explosive and great smells, they stopped him and they strip searched him 20 times over because he was covered in what, by the reckoning of the machine, were explosives.
Yeah.
So that shows you that there's plenty of scope, but also the machine that will be able to tell all different explosives is not there yet.
Yeah, well, I guess in terms of sensors, there's nothing like biology. Right?
Yeah, Clifton Malone, who's a great analytical chemist, who worked on isotopes, on the different smell of isotopes. He used cockroaches to test the smell of isotopes. And he got amazing results. The cockroaches could smell deuterated Anyway, he told me that he got into smell because a student of his pointed out that our nose was much better than any analytical instrument that they had at their disposal. And Clifton, being an analytical chemist, took this rather personally and worked on smell for the next decade.
Well, Luca, this has been a fascinating conversation. I've been meaning to talk to you for a while, so I'm glad this worked out. And I would encourage my listeners to, I guess, dig in, use AI to understand our conversation, because there's a lot in there that you mentioned that is learnable. I mean, to be honest, I didn't even remember what an isotope was. I had to go refresh my memory. Isotopes, isomers, topomers. So I'd encourage the listeners to dig in and look into these. But do you have any advice? I mean, I usually end my conversations by asking people, what kinds of advice they have for young people. In this case, for people studying smell, or life in general. How would you advise people to?
So life in general, my advice would be don't do what I did.
Well, I'm not sure I believe that.
But no, one absolutely revolutionary thing that's happened in the field of perfumery is that it used to be the most, I suppose this side of the circus, it used to be the most closed industry. It was father to son or father to daughter within a very small clan of people.
Yeah.
Now, independent and artisan perfumers are cropping up by the hundreds.
So is this because of the internet?
Yes. Because of the internet, it's because the possibility of direct sales.
Yeah.
So without going through a brand, you just have your little web shop and you sell perfumes and people buy them.
Yeah.
And paradoxically, it's also due to the price inflation. Perfumes are quite expensive these days. So let's say if you're going to go into a duty free and buy a perfume for $200, if you give $200 to an independent perfumer that doesn't have all the advertising costs and all the packaging and all that stuff, it goes in the formulas. Mm-hmm. So the perfume smells great if it's well done. So there's a rise. All of a sudden, hundreds, thousands of people are getting into perfumery because they didn't read the memo that said you can't do that.
Yeah.
Okay. Yeah. And some of their work is amazing. Okay. So for example, the stuff I'm wearing today, which is called Bestia Sacra by a Russian guy who's in Thailand, Maxim Bortnikov, his name is a fantastic perfume. The guy is self-taught. Actually, his father also taught him. But this is a revolution. So my advice to people who are into smell and perfumery is start playing around. Get yourself some aroma chemicals and have a good time.
Yeah.
Sunday painters. What we need is hundreds of Sunday painters.
When I was researching you, I came across someone who said you recommended a perfume by a perfumer called the Zoo.
Yes.
And I think it was Everlasting or something.
Yeah, fantastic.
Am I remembering right? So I looked it up, and it's out of stock. Yeah. So you obviously had an impact on the sales of that.
I think I did. In fact, I've made a vow never to praise a perfume that you can find on eBay until I've bought a good stock of it for myself, because I've shut myself out of a number of really good deals.
Yeah.
Can I make a plug for my Substack?
Yeah.
I have a Substack in which I discuss, among other things, perfume, mostly perfume, but also weird stuff, including long enthusiastic rants about AI.
Enthusiastic.
Yeah, very positive.
Yeah.
And it's lucaturin.substack.com.
Very good. Well, Luca, on that note, thank you so much. It's been delightful talking to you, and I look forward to our next conversation.
Yeah, same here. Thank you so much.
All right.